Okay, I'll start now. So, hello everyone. Uh, I'm Shrija Ghosh, a PMRF from IIT Kanpur, and I'm taking the problem solving sessions for the course Environmental Chemistry and Microbiology. Today is the 4th week session, and the course instructors for this course are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur. So, in this session, we will be discussing different questions related to different kinds of catalysts and their mechanisms, and we'll see how a catalyst can help change the rate of the reactions. So, the chemical kinetics that we have studied in the till the last session, we'll see how some of the applications of catalyst in that area. So, I'll start from the first question. Which is incorrect for a catalyst? So, we have 4 options. The first is a catalyst remains unchanged in mass and chemical composition at the end of the reaction. Second option is it does not alter the position of equilibrium in a reversible reaction. Third is a catalyst can initiate a reaction. And the fourth option is coenzymes increase the activity of an enzyme. So, we have to select the incorrect option. So, can anyone answer which is the incorrect option from all this? Yes, so option number C is the incorrect option. So, a catalyst remains unchanged in mass and chemical composition at the end of the reaction. This is correct answer because in any catalyst, in any catalyzed reaction, we always get the catalyst back in the catalytic cycle. So, when a reaction is going on, the reactants will interact with the catalyst, but at the end the catalyst will be regenerated. So, it does not, the mass of the catalyst remains unchanged. It does not get consumed in the reaction. Okay, it gets replenished in each catalytic cycle. So, the first option is correct. Second option, it does not alter the position of equilibrium in a reversible reaction. So, what a catalyst does, catalyst simply changes the rate of the reaction. It cannot alter anything other than the rate of the reaction. It just lowers the activation energy. It can change the path of the reaction, but it cannot alter the position of the equilibrium in a reversible reaction. It can only make it go faster or slower. Okay, so the second option is also correct. Third option, a catalyst can initiate a reaction. This is the incorrect statement because a catalyst can never initiate a reaction. Okay, a catalyst can never initiate a reaction. Reaction will be initiated by the reactants and the products. The catalyst, what can a catalyst do? A catalyst can only make the reaction either go faster or slower. Okay. The fourth option, coenzymes increase the activity of an enzyme. It is a correct statement because coenzymes are basically helper enzymes. So that which is so enzymes are kind of biocatalysts. It catalyzes the biological reactions that goes on in our body. So coenzymes are the factors that can help those enzymes function in a better way. Okay. Just a second. So the correct answer for this question is the option number C. Catalyst does not initiate the reaction, it simply increases or decreases the rate of the reaction without itself being consumed at the end of the reaction. That's why the first option was also like correct statement for a catalyst. It remains unchanged in mass and chemical composition. Okay. So moving on to the second question. To maintain the strength of H2O2, hydrogen peroxide, what type of catalyst are you using? So, to maintain the strength of H2O2, what type of catalyst will be required? Anyone wants to answer this question? So, let's see what all options are there. We have positive catalyst, negative catalyst, auto catalyst and none of the above. So, positive catalyst, what do you mean by positive catalyst? Positive catalyst means something which can increase the rate of the reaction. So, positive catalyst here means it can increase the rate of the reaction. Okay. Negative catalyst means it can decrease the, it's just opposite of positive catalyst. So, it can decrease the rate of the reaction. Autocatalyst means some product that is forming which acts as the catalyst for the reaction itself. So, in this case, to maintain the strength of H2O2, we have to make sure that the H2O2 does not get degraded. H2O2 gets decomposed into H2O plus O2, water and oxygen molecule. So, we have to make sure that that reaction is slowed down. So, to slow down the reaction, we need a negative catalyst. So, option number B is the correct answer. So, strength of H2O2 decreases with time gradually as H2O2 will decompose into water. So, H2O2 will get converted into H2O plus O2. Okay. So, we use a negative catalyst which will slow down the process of decomposition. Okay. So, to maintain the strength, so if we want the amount of H2O2 to be constant in a bottle, we have to add a negative catalyst so that it can stop the decomposition of H2O2 into water and oxygen. Okay, this is clear. Moving to the next question. Question number 3. Which of the following is an example of an autocatalyst? So, I have already told you right now what is an autocatalyst. Autocatalyst means a product that is forming in the reaction which acts as the catalyst itself for its own reaction. So, let us say I have a reaction A goes to B and let us say B is the catalyst, B can act as a catalyst for this reaction itself, A goes to B. So, this will be a case of autocatalyst. In this case, we do not have to add an external catalyst. Okay. So, we have 4 options. First is finely divided iron in ammonia synthesis by Haber's process. Phosphoric acid in the decomposition of H2O2, second option. Third option is redox reaction between potassium permanganate and oxalic acid. Fourth option is palladium for hydrogenation of ethylene. Okay, all of these are catalyst driven reactions, but which of the reaction is actually an autocatalyst kind of reaction? So, in this case, the correct answer is option number C. Okay. In the first reaction, finely divided iron in ammonia synthesis by Haber's process, it is a positive catalyst type of reaction. Why positive catalyst? Because the finely divided iron, it acts as the catalyst to increase the rate of the reaction so that ammonia is synthesized in a quicker way. Okay. So, iron increases the rate of the reaction. Similarly, the last option, palladium for hydrogenation of ethylene, this is again a positive catalyst type of reaction, because palladium acts as a positive catalyst and it can speed up the reaction of hydrogenation of ethylene. So, ethylene gets hydrogenated to form ethane. Okay. So, palladium also acts as the positive catalyst in this case. The second option, phosphoric acid in the decomposition of H2O2, we right now discussed, it's a negative catalyst. So, it stops the decomposition of H2, it slows down the decomposition of H2O2. So, it acts as a negative catalyst. Okay. The third option, redox reaction between potassium permanganate and oxalic acid. This is the correct answer. This is an example of autocatalyst. So, let us see what is happening in this reaction. So, in this redox reaction between potassium permanganate and oxalic acid, so we have MnO4 minus, which is basically KMnO4, potassium permanganate is KMnO4. So, if you write it in ionic form, it will be MnO4 minus plus 16H plus 5C2O4 2 minus. So, oxalic acid will be in ionic form, it will be oxalate, and this will get converted into 2Mn2 plus 8H2O plus 10CO2. So, what happens? It's a famous reaction that is being done to determine the concentration, to do the titration of oxalic acid basically. So, potassium permanganate has a bright pink color which gets neutralized with oxalic acid and the color disappears. So, initially when few drops of oxalic acid is added, the color does not disappear immediately. What happens? After few drops of oxalic acid is added, some of the MnO4 minus gets converted into Mn2 plus. This Mn2 plus acts as the catalyst for the reaction, and then once few small amount of Mn2 plus has already been formed, it acts as the catalyst and it can catalyze this reaction at much faster rate, and the reaction proceeds towards the RHS. Okay. So, here in this case no additional catalyst is added, the product, one of the product, that is Mn2 plus, it acts as the catalyst itself. Okay. I hope autocatalysis was clear. Is there any doubt in this question or in this reaction? Okay. So, we will move on to question number 4. The role of As2O3 in the contact process of H2SO4 synthesis is. What is the role of As2O3 in the contact process of H2SO4? So, in the contact process, so we have 4 options again: negative catalyst, positive catalyst, catalyst poison and catalyst promoter. So, now in the contact process of H2SO4 synthesis, what does As2O3 does? What As2O3 does is, it acts as a catalyst poison. Okay. So, negative catalyst, I told you, it means something that can decrease the rate of the reaction, that can slow down the reaction. Positive catalyst, something that can increase the rate of reaction. But what is catalyst poison? Catalyst poison means a third compound that can degrade or that can stop the catalyst to function. So, it is an inhibitor of a catalyst itself. Okay. So, do not get confused with negative catalyst and catalyst poison. Okay, negative catalyst means certain compound X that can decrease the rate of the reaction A goes to B. But catalyst poison means some other compound Y, it can actually act on X and stop X from increasing or decreasing the rate of the reaction. Okay. So it will be a poison for the catalyst, and catalyst promoter, it's the opposite of catalyst poison, something that can promote the action of a catalyst. Okay. So in the contact process of H2SO4 synthesis, As2O3 destroys the catalytic activity of platinum catalyst. So in this H2SO4 synthesis process, platinum is used as a catalyst, but As2O3 can destroy the catalytic activity of platinum. So it acts as the catalyst poison or anti-catalyst. It is also known as anti-catalyst. Okay. I hope this is clear. What is the difference between negative catalyst and catalyst poison? Anyone has any doubt? Okay. So we'll move on to the next question. Question number 5. An exothermic reaction with high activation energy for the forward reaction is given by which of the diagram most appropriately? So it's an exothermic, so we have two information in this question. First is it's an exothermic reaction. Second is with high activation energy. Okay, high activation energy for the forward reaction. So what do you mean by exothermic reaction? I have told you in the previous sessions also. Exothermic reaction means, anybody?
The reaction in which heat is evolved.
So the reaction in which the heat is released, the heat is expelled out of the system. So if in a reaction the heat is being released or the energy is being released, that means the products will be at a lower energy state than the reactants, or the reactants will be at a higher energy state than the products. Okay. So if you see in the first case, the reactant is at a lower energy than the product. So this is not an exothermic reaction, because energy is required to make the product, because product is at higher energy. Okay, so it is a case of endothermic reaction. For the second case also, the reactant is at a lower energy state than the product. So second option is also not an exothermic reaction. Third and fourth, both options are an example of exothermic reaction, because reactant is at a much higher energy state than the product. So energy will be released while the conversion of reactant into the products. Okay. So C and D, both options are of exothermic reaction. Now the second information that they have given in this question is high activation energy. Okay. So let's see, in option number C, the activation energy, you can see, what do you mean by activation energy? Activation energy means the energy required by the reactant to reach the transition state, after which the reaction will proceed towards the product. So that the highest point of this curve, the tip of this curve, it will be the energy difference between the reactant's energy and the maximum energy attained, that is the activation energy. Okay. So now if you see in the plot C, the activation energy is lower, because the difference between the topmost point and the energy of reactant is not that is not very significant. But in case of option number D, you see the difference between the reactant's energy and the energy uh, of the maximum, the top point of this curve, it is very high, it is significantly high. So the option number D is showing an exothermic reaction with a high activation energy. Okay. So in this case, option number D is the correct answer. For exothermic reaction, the reactants are at a higher energy level compared to the product. Okay. Okay, so this is clear. Okay. So moving on to the next question, question number 6. The following diagram shows the energy diagram for a chemical reaction. The point on the diagram representing the transition state is. Now again we have 4 points given, A, B, C and D. Which point is showing the transition state? Anybody wants to answer this question? Yeah. Yeah, the option number B is the correct answer. So, what do you mean by the transition state? So, you have a reactant and you have a product. Okay. When the reactant is getting converted into product, it does not directly gets converted. It goes through a certain energy level at a higher energy state where a transition state is formed, and from that higher energy state it comes back to the lower energy state wherever your product energy is there. Okay. So if I asked if this reaction curve is a endothermic reaction curve or a exothermic reaction curve, what do you think? Based on the previous question, what do you think, that if A is the reactant and D is the product, what do you think, if it whether it's a exothermic reaction curve or an endothermic reaction curve?
Endothermic.
Yes. Why it is an endothermic reaction?
Energy is required in case of endothermic reaction.
Yeah, because A is at a lower energy state than the D. So to make D from A requires energy. Okay, because D is at a higher energy state. So that means this is an endothermic reaction, and B is the transition state of this reaction, the topmost point. Okay. So the transition state is the highest energy species that exists during the course of a chemical reaction. Okay. In the case of this reaction, the highest energy point is B, hence this must be the point where the transition state exists. So basically the transition state is the highest energy species. So in any curve, the topmost point will be your transition state energy. Also in this curve, activation energy, if we have to see the activation energy will be the energy difference of A minus the energy difference of B. So the difference between the energy states of A and B, that is the activation energy. So in this reaction curve, A requires a lot of energy to reach to B in order to get converted into C and D. Okay, C must be a intermediate in this case. Okay. So moving to the next question, question number 7. In the presence of sodium sulfite and oxygen, AS3 arsenic in the oxidation state of 3 will be oxidized to AS5, okay, oxidation state 5, and sodium sulfite also gets oxidized into sodium sulfate. This is an example of. So, what is the reaction basically? Sodium sulfite and arsenate gets oxidized into sodium sulfate and arsenite gets oxidized into sodium sulfate and arsenate. Okay, arsenic also gets oxidized from oxidation state of 3 to oxidation state of 5 plus 5, and sodium sulfite also gets oxidized into sodium sulfate. So, sulfite SO3 minus gets oxidized into SO4 2 minus. So, this is an example of induced catalysis. Why induced catalysis? Because if you see only the oxidation of arsenite into arsenate, AS in oxidation state 3 plus into AS in oxidation state of 5 plus, that reaction is very slow. But when we induce this reaction with another reactant which also gets oxidized along with the arsenite, in that case the reaction goes at a much faster rate. So, Na2SO3 acts as the inducer of this reaction. So, it acts as the induced catalyst. Okay. So, this reaction is an example of induced catalysis. Na2SO3 plus Na3ASO3 plus O2 gives Na2SO4, that is sodium sulfate, plus Na3ASO4. Okay. Is this clear? Is there any doubt till this point? In any of the previous questions as well?
Would you please repeat the answer of this question number 7, please?
The question number 7, the answer is induced catalyst. Option number C. So, in this case, what is happening is AS in oxidation state 3 plus, it is getting oxidized into AS in oxidation state of 5 plus. Okay, but this oxidation alone is very slow. So, if we add another reactant, that is Na2SO3, so it kind of induces the oxidation of AS3 to AS5. So, Na2SO3, it also gets oxidized with the oxygen. So, it becomes from Na2SO3 it becomes Na2SO4, from sulfite to sulfate, and along with that it also lets the oxidation of arsenite to arsenate occur at a much faster rate. So, Na2SO3 is inducing the catalysis. It acts as the induced catalyst. Okay. So, option number C is the correct answer. Clear?
Yes, yes.
Okay. We have a question. Ma'am, what is the difference between induced and enhancer catalyst? So, in case of enhancer catalysis, just a second, enhancer catalysis means positive catalysis. Okay. Yeah. So enhancer catalysis means positive catalysis. So in case of positive catalysis or enhancer catalysis, you have a catalyst molecule which can increase the rate of the reaction. In case of inhibitor catalysis, you have something that can decrease the rate of the reaction. But induced catalysis, it can how do I say, it can also increase the rate of the reaction, but it is also getting oxidized in this process. Okay, it is also a part of the reaction. Okay. So in case of induced catalysis and autocatalysis, in both of these cases, external catalyst is not added. It is a part of the reaction. So in case of autocatalysis, how I explained that the product of the reaction will act as a catalyst and it will speed up the reaction, speed up its own reaction. Okay. In case of induced catalysis, it is it is one of the reactant that is acting as the catalyst. Okay. Yeah. Okay. So moving on to the next question. In Chambers' process of sulfuric acid synthesis, nitrogen oxide is used as a catalyst. It is an example of. So, Chambers' process of sulfuric acid synthesis, in this case, nitrogen oxide is used as a catalyst to synthesize sulfuric acid from SO2 and water vapor, H2O and SO2 combine to form H2SO4 in presence of nitrogen oxide. And we have to tell what is the type of the catalysis happening. So we have four options: heterogeneous catalysis, homogeneous catalysis, biocatalysis and micellar catalysis. So what is heterogeneous catalysis? Heterogeneous catalysis means the physical state of the catalyst and the reactants and the products are different. So if the catalyst is solid and the reactants and products are liquid or gaseous, that is an example of heterogeneous catalysis. In case of homogeneous catalysis, the state of all the reactants and the catalysts are same. So catalyst, reactants and products, everything is in gaseous phase or everything is in liquid phase. That is an example of homogeneous catalysis. Biocatalysis means it is the reaction is catalyzed by an enzyme. Okay, enzymes are proteins that can catalyze, that can speed up some biological reactions. Okay, so biocatalysis means it is catalyzed by an enzyme, and micellar catalysis means it is catalyzed by a surfactant or a detergent that can form micelles. Okay. So in this case, nitrogen oxide is the catalyst. So it is not a biocatalysis and it is not a micellar catalysis because it's not an enzyme or a surfactant. Nitrogen oxide is not an enzyme or a surfactant. Now, it is an example of a homogeneous catalysis because the state of the nitrogen oxide and the reactants, the SO2 and water vapor, that is everything is in gaseous state. So that's why it is an example of homogeneous catalysis. Okay. So moving on to the next question. Paul Anastas and John Warner developed 12 principles of green chemistry in 2000. Which statements among the following is or are true? So, John Warner and Paul Anastas developed 12 principles of green chemistry, and we have to tell which of the following statements are part of that principle. So first statement is analyze processes in real time. Second is use renewable feedstock. Third is prevent waste, and fourth is all of the above. So which option is the correct option for this question? If anyone wants to answer, it's a simple question. Anybody? Yes, option number D is the correct option, because all of these statements are part of the 12 principles of green chemistry. So green principles of green chemistry was developed to uh, like basically it was defined to make certain amendments in order to make a sustainable environment. Okay, so if we see the answer is all of the above. And if we see what are the 12 principles of green chemistry, first is prevent waste. We need to prevent waste production in order to make a sustainable environment. Okay, in order to make a cleaner environment. Second is design safer chemicals and products. So all the chemicals that have been used in the industry after ever since the industrial revolution, these chemicals, like many of these chemicals, are potent pollutants of the environment. Okay, and most of the wastes are disposed of in the water bodies after minor treatment. So mostly these pollutants, these polluting chemicals, they go into the water body and at the end affect the groundwater and the and that water only evaporates and goes in our atmosphere. So it is affecting the atmosphere as well as the ecosystem of the planet. So design safer chemicals and products. Third is design less hazardous chemical synthesis. So nowadays what people are trying, people are more and more getting oriented towards green chemistry. They are trying to synthesize all these commercially available and commercially important chemicals in a less hazardous way. So they are designing different schemes, different mechanisms, so that these chemicals can be synthesized in a less hazardous way. Okay, so that less and less pollutants are being released as a waste. Fourth is use renewable feedstock. What do you mean by renewable feedstock? The feedstock that we are using for synthesis of different chemicals, those feedstock, if they are non-renewable, like till now we all are using petroleum as our basic requirement, but petroleum, as we all know, it's non-renewable resource. So after a certain point it will get depleted sooner or later. So if we can switch to a renewable feedstock, we can make a environment which can last longer. Okay. Fifth is use catalysts. So why we are promoting catalysts so much? Catalysts can increase or change the rate of the reactions. Like some reaction, if we want it to occur faster, it can make it happen. And on the same point, people have been switching to biocatalyst, because naturally if something is present in the biological system, it must be biodegradable and it will be less harmful to the environment. So people have been promoting biocatalysts in this, because of this. Sixth is avoid chemical derivatives. So again, chemical derivatives in a like in any like let's say if someone is making drugs or anything, like if they have more and more chemical derivatives, it will remain in the environment, it won't be biodegradable. So that's why people try to switch towards biological uh, products and biological entities, so that it can be it will be biodegradable in the nature. Seventh is maximize atom economy. Eighth is use safer solvents and reaction conditions. Ninth is increase energy efficiency. 10th principle is design chemicals and products to degrade after use. 11th is analyze processes in real time to prevent pollution. And 12th is minimize the potential for accidents. So all these principles are part of green chemistry, which were developed by Paul Anastas and John Warner. Okay. So moving to the next question, question number 10. Which of the following is an example of heterogeneous catalysis? First is acid hydrolysis of cane sugar to form glucose and fructose. Second option, active charcoal in the formation of phosgene from CO and Cl2. Third is trans-esterification of fatty acid triglyceride with methanol. And fourth is all of the above. So what is heterogeneous catalysis? I have already told you. Heterogeneous catalysis means where the reactants and the reactants or products and the catalyst that is being used are having different physical states. So if the reactants are liquid and the catalyst is also liquid, it is an example of homogeneous catalysis. Okay. So in this question, the correct answer for this question is option number B, active charcoal in the formation of phosgene from CO and Cl2. So phosgene is a gas, uh, phosgene is a gas, COCl2. It is formed from carbon monoxide and chlorine gas. So all the reactants and the products are in gaseous state. But active charcoal, it is in solid state. So it is an example of heterogeneous catalysis because the catalyst is in a different state than the reactants and the products. Okay. The first option, acid hydrolysis of cane sugar to form glucose and fructose, it is an example of homogeneous catalysis because everything is in liquid state. Trans-esterification of fatty acid triglyceride with methanol, this is also an example of homogeneous catalysis because everything is in same state. Okay. So the correct answer is active charcoal in the formation of phosgene from CO and Cl2. Okay. Clear till this point? If anyone has any doubt, they can ask or they can write in the chat box. From question number 1 to 10, is there any doubt? Okay, then I'll move to the next question. Question number 11. 40 mg per liter of a textile dye, which having a molecular weight of 408 grams per mole, is completely degraded catalytically. The catalyst was used at a dose of 0.8 grams per liter and the time taken was 1.5 hours. Calculate the turnover frequency of the catalyst. So now what is turnover frequency? Turnover frequency is nothing but turnover number per unit time. And now what is turnover number? So turnover number, it differs for homogeneous and heterogeneous catalysis, but the idea is same. The turnover number basically means the number of molecules of the substrate that is being catalyzed per molecule of catalyst or per gram of catalyst. Okay. So in this case we are talking about per gram of catalyst. So the amount of catalyst is already given, 0.8 grams per liter. We have to first calculate the number of molecules of the textile dye that is being degraded. So textile dye is the substrate here. So we have to find the number of molecules of this dye that is being degraded. So complete degradation is happening of 40 mg per liter of dye using 0.8 grams per liter. So first we will have to calculate the number of molecules of this dye that is being degraded. Once we have the number of molecules, then we have to find the turnover number by calculating the number of molecules catalyzed per gram of catalyst. So by 0.8 grams per liter catalyst, whatever number of molecules are getting degraded for the textile dye, you have to divide it by 0.8 in order to find the per gram, in order to find the number of molecules per gram of catalyst. Okay, and then we will divide it by the time taken in order to find the turnover frequency, because turnover number is just the number of molecules of the substrate degraded per or catalyzed by a catalyst per gram of catalyst. Okay, but turnover frequency means turnover number per unit time. Okay. So how do we calculate number of molecules for any substance? First we will have to calculate the number of moles. Okay. So 40 mg per liter, and we have the molecular weight also given. So we will just convert it into we will just convert the mg into gram, and then we can divide the given mass and the molecular mass. But how will we get the number of molecules? So 1 mole has how many molecules? Anybody? 1 mole of substance. How do we define the number of molecules for a 1 mole of substance? Yes, 6.02 into 10 raised to power 23. This is the Avogadro's constant. Okay. So 1 mole of molecule has 6.02 into 10 raised to power 23. 1 mole of substance has 6.02 into 10 raised to power 23 molecules. So what is the number of moles we are getting for this dye? So we have 40 mg per liter. So 40 mg means 40 into 10 raised to the power minus 3 grams per liter divided by the molecular weight 408 multiplied by 6.02 into 10 raised to the power 23. So this will be molecules per liter. Okay. So 0.8 gram is catalyzing the reaction for this many molecules. So 1 gram of catalyst will catalyze how many molecules of reaction? So it will be 40 into 10 raised to the power minus 3 into 6.023 into 10 raised to the power 23 divided by 408 divided by 0.8 grams per liter. So that will be the turnover number. but we have been asked to find the turnover frequency. So to calculate the frequency, we have to consider the time factor. So turnover number per unit time is the turnover frequency. So what is the time taken? It was 1.5 hours. So 1 hour has 60 minutes and 1 minute has 60 seconds. So 60 into 60 is 3600 seconds. So 1.5 into 3600 will be 5400 seconds. Okay. And now we can just solve everything. 40 into 10 raised to the power minus 3 into 6.023 into 10 raised to the power 23 divided by 408, which is the molecular weight, multiplied by 0.8, which is the dose of the catalyst, multiplied by 5400, which is the time taken. Okay. So you will get the answer in molecules per gram per second. So molecules per gram, number of molecules of the substrate catalyzed by 1 gram of catalyst per second is the turnover frequency. Okay. So the answer we are getting here is 1.37 into 10 raised to the power of 16, which is option number C. So option number C is the correct answer. 1.37 into 10 raised to the power of 16. Okay. I hope this calculation is clear. Okay. So we'll move on now to the next question. Okay, so question number 12. Which of the following is used as a catalyst in Chambers' process of sulfuric acid synthesis? So I have already discussed this in the previous question. In the previous question it was already given. So in Chambers' process of sulfuric acid synthesis, nitrogen oxides are used as the catalyst, and it was the type, what type of catalyst it was? It was a homogeneous catalysis. Okay, because everything was in gaseous state. Nitrogen oxide and the reactants, everything was in gaseous state. So it was a homogeneous catalysis, and the catalyst uses nitrogen oxides. Okay. So this is something we have already discussed. Now the question number 13. If a compound X with the molecular weight 139.11 grams per mole, having concentration 10 micromolar, is reduced completely in the presence of catalyst at a dose of 10 grams per liter, then calculate the turnover number of the catalyst approximately. So it is very similar to the question number 11 that we solved just now. So we have a compound X, we have the molecular mass, molecular weight given, and here we have the concentration in terms of molar. So 10 micromolar is the concentration. Catalyst dose is given as 10 grams per liter. Okay. So now from the concentration, first we have to find the number of molecules. So from the concentration given, 10 micromolar means 10 into 10 raised to the power of minus 6 moles per liter. Okay, and that multiplied by the Avogadro's constant will be the number of molecules. Okay, so in this case we have to find the turnover number, not the frequency, because they haven't given the time factor. So 10 into 10 raised to the power of minus 6 into 6.023 into 10 raised to the power of 23. That will be the number of molecules per liter. And the dose of catalyst per liter is given as 10 grams per liter. That means 10 grams of catalyst can catalyze the reaction of these many molecules. Then what will be the turnover number? Turnover number is basically how many molecules are catalyzed by 1 gram of catalyst. So if this is the number of molecules that is catalyzed by 10 gram of catalyst, for 1 gram of catalyst it will be divided by 10. So 10 into 10 raised to the power of minus 6 into 6.023 into 10 raised to the power of 23 divided by 10. So it will be, if you solve this, 10 and 10 will get cancelled and 23 minus 6 is 17. So 6.023 into 10 raised to the power of 17 molecules per gram of catalyst. Okay. So the option number C is the correct answer, 6.0 into 10 raised to the power of 17 molecules per gram. Okay, for question number 13, option number C is the correct answer. Okay, this is clear how to calculate the turnover number and the turnover frequency. So let's say they had given the time also, so we have to divide the turnover number by the time to get the turnover frequency. Okay. So moving on to the next question, question number 14th. Which theory gives a proper explanation of the mechanism of heterogeneous catalysis? So heterogeneous catalysis, what did I tell you? Heterogeneous catalysis is where the catalyst and the reactants and the products have a different physical state. So if the catalyst is solid and the reactants and products are liquid or gaseous, it will be an example of heterogeneous catalysis. So what is the theory that explains the mechanism of heterogeneous catalysis? So the options are absorption, adsorption, both of the above or none of the above. So the theory that explains the mechanism of heterogeneous catalysis, it is the adsorption theory. Okay. What do you mean by adsorption? Adsorption is generally defined in terms of solid surfaces. So on any solid surface, any liquid or gas, it kind of sticks to the solid surface, and that phenomena is called adsorption. Okay. So I have an example of this adsorption theory also. So see this reaction. It is hydrogenation of ethylene to form ethane. So C2H4, that means a double bond is there. Okay, carbon-carbon double bond is there, and then two hydrogens are attached to each carbon. C2H4 plus H2 gives C2H6. So this reaction itself is a very slow reaction. But in presence of catalyst, here the catalyst is platinum. So all the reactants and products, you can see it's in gaseous phase. But the catalyst platinum, it is solid. Okay. So now you have a solid, imagine a solid plate kind of thing. Okay. So when you are just adding the reactant C2H4 and H2, since these are gas molecules, these are very spread apart, and the chances of the molecules of C2H4 colliding with the H2 for the reaction to occur is very less, because they both are gases and the molecules are very far apart of each other. But when you add this catalyst platinum, since it's a solid, what happens is the C2H4 and H2, they get adsorbed on the platinum surface. Okay, the hydrogen atoms, the H2 dissociates into hydrogen atoms, and each hydrogen atom gets attached to the platinum surface, and C2H4 gets attached to the platinum surface, and each of the H2 atoms then now once they are attached to the attached to a solid surface, they are less mobilized. Okay, when they are free in the gaseous state, they are very mobile, so they can move here and there, but once they are fixed on a solid surface, now there is the chances of them colliding and the reaction occurring is faster. So what happens, once the reactants have adsorbed on the catalyst surface, the hydrogen now can attack the ethylene molecule to form ethane. Okay, and once ethane is formed, since it's a gas, it will be released from the platinum surface, and the platinum surface will be again ready for the next cycle of reaction, and that's how a catalyst keeps on getting regenerated, so it does not get consumed in the reaction. Okay, so I hope you understood the adsorption theory of heterogeneous catalysis. So in case of heterogeneous catalysis, since mostly the catalyst is solid state and the reaction is a liquid or a gaseous state reaction, the adsorption theory holds well for the for this type of catalysis. Okay, so the question was, which theory explains the mechanism of heterogeneous catalysis? The answer will be adsorption theory. Okay, so coming to question number 15. If the melting point of bulk gold is 1064 degrees Celsius, then the melting point of gold nanoparticles may be. So we have the melting point of bulk gold as 1064 degrees Celsius. What will be the melting point of the gold nanoparticles? What is nanoparticle? Nanoparticles are basically particles whose any one dimension of it will be in the range of 1 to 100 nanometers. Okay, any one dimension. So if you have gold nanoparticle, what will be the melting point? Will it be the same as the bulk gold? It will be greater than 1064 degrees Celsius, or it will be very low? What will be the melting point? So for any nanoparticle, the properties of the nanoparticles are very different from the properties of the bulk material. So both of them are gold, but since it's a nanoparticle, the properties change drastically. So in this case, the melting point of the gold nanoparticles is very low. It may be even less than 23 degrees Celsius. Okay, so the answer is option number C, very low. So you can see the drastic change. The bulk gold, it had a very high melting point, 1064 degrees Celsius, whereas the gold nanoparticles has a very low melting point. It can be less than 23 degrees Celsius. Okay, so there is a huge difference between the properties of nanoparticle from their own bulk material. Okay, okay, moving to the next question. Question number 16. Detergent and soap in water has some common properties. Both of them are. So detergent and soap, these are all surfactants. Okay, surface active agents. So surfactants. So what property do they have in common? Option number A is biodegradable in nature. Option number B is small hydrophobic molecules. C is suitable for decreasing the surface tension of water, and D is suitable for increasing the surface tension of water. So the answer is option number C, suitable for decreasing the surface tension of water. See, first option is not correct because soaps are not biodegradable in nature. They remain in nature, they are not biodegradable. Second option, you must be thinking second option can be correct, but if you see a structure of a detergent or a soap, they have both hydrophobic and hydrophilic parts. So detergents are generally long chain molecules, and one end of that chain is hydrophobic but other end of that chain is hydrophilic, and that's how they get dissolved in the water. Okay, if they were completely hydrophobic, they won't dissolve in water at all, they will form a layer above the water. But after a certain concentration of detergent, when you mix detergent or when you mix soap in water, it does dissolve in water. Why does it dissolve in water? Because they have hydrophilic ends that can interact with the water molecule. Okay, but when you mix soap in water, you must have seen the bubbles forming. Okay, those bubbles form because the surface tension of water gets decreased. Okay, so both detergent and soap, they act as surfactants, and the surfactant molecule decreases the surface tension of water. Okay, so once the surface tension of water is decreased, the water can, that's why those bubbles form. The bubbles basically they are spheres of the water in the detergent water and the detergent molecule with air inside it. Okay, I hope this was clear. So moving to the next question. Question number 17. Sodium dodecyl sulfate is. So what is sodium dodecyl sulfate? Does anybody know what is sodium dodecyl sulfate? If you would have used it in your lab, it's a very common reagent. Yes, it's an anionic detergent. It's very commonly used in protein gel electrophoresis actually. So sodium dodecyl sulfate, it's a anionic surfactant. So we had four options: cationic surfactant, anionic surfactant, zwitterionic surfactant, and non-ionic surfactant. Yes, it is used in SDS page, correct. So sodium dodecyl sulfate is basically, it's an anionic surfactant, and here is the structure of the sodium dodecyl sulfate. So how do we determine whether it's an anionic detergent or a cationic detergent? So you can see the structure has two ends, one CH3 end and one is this SO4 minus. So if you see sodium, so once you dissolve SDS in water, the sodium will get dissociated like any ion. When you dissolve NaCl in water, Na plus and Cl minus separates out. Similarly here Na plus and the sulfate minus part will separate out. But in this sulfate SO4 minus, you can see this minus sign. So this is an anionic end. Okay, this is a hydrophilic end, but it is anionic in nature. It is negatively charged. That's why the whole surfactant is called the anionic surfactant. If it was a cationic surfactant, there would be a positive, some positive ion at the end of the chain, and it will be having positive charge. What do you mean by Zwitter ion? Can anyone answer? What do you mean by Zwitter ion? Yeah, dual ion. You are partly correct. So basically, Zwitter ion as, yeah, so you in the structure, in the anionic structure, you will have both positive and negative charges. Yes, exactly, zero valent. This is the correct definition. So the overall charge of that molecule will be 0. It will be an uncharged molecule, but it will have both positive and the negatively charged moieties. Okay, so it's a very common term used in terms of amino acids. So amino acids, when they get when they start getting deprotonated at certain stage, you can have a CO minus and NH4 plus at the same time in an amino acid structure. So in that case, the net charge on that amino acid will be 0, and it will be an Zwitter ionic compound. So similarly, in any surfactant, if you have a side chain, sorry, not side chain, if you have one end, the hydrophilic end, having both positive and negative charge, like here in this case SO4, one of the O has negative charge. Let's say some other group was there where one of the group had positive and one of the group had negative charge. So in that case, it would have been a Zwitter ionic surfactant. And by non-ionic surfactant, it's very clear, you have something which is not ionic at all. It is not charged. It is an uncharged molecule. Okay, so there is a difference between, so both Zwitter ionic and non-ionic surfactant will have charge 0, but Zwitter ionic surfactant will have both positive and negative charge present. The net charge will be 0. For non-ionic surfactant, there will be no charge present on the molecule itself. Okay, so this is the major difference between the Zwitter ion and the non-ionic surfactant. Okay, so next question. So next question, question number 18. Surfactant molecules can form micelle in water medium when the surfactant concentration is. So I have already told you about detergents. When detergents are dissolved in water, since they have a hydrophilic end and a hydrophobic end, like in the structure I have shown you previously for SDS, we have the CH3, the carbon chain, which is hydrophobic, and the anionic part which was hydrophilic. So those surfactant molecules, when they are dissolved in water, after certain concentration they form micelles in water. What are micelles? Those are kind of spherical structures, globular structures you can say, where all the hydrophobic, like if it is in water, all the hydrophobic ends will get concentrated towards the center of the sphere, and the hydrophilic ends will be towards the surface of the sphere, which facilitates the solubility of that surfactant molecules in the water. So the hydrophilic end can interact with the water medium happily, and it will make the surfactant molecules stay inside the water medium despite of having an hydrophobic end. Okay, all the hydrophobic end will get concentrated towards the center of that sphere. So that sphere is called micelle. So that micelle formation occurs only after a certain concentration, which is called critical micellar concentration, CMC. Okay, so the first option above the CMC of the surfactant, it is the correct option. Once that particular concentration of surfactant is added, only then it will be dissolved in water medium. So only then it will form micelle. So before that, if you add very small amount of surfactant below its critical micellar concentration, it will not form the micelles inside the water medium. Okay, so surfactant molecules form micelles in aqueous medium above the critical micelle concentration, that is CMC, for that surfactant. Okay, so every surfactant has a different CMC value, different concentration of different surfactants are required to form the micelles. Okay. I hope this was clear. We'll move to the next question. What must happen before a chemical reaction can start? So, I have discussed the energy diagram of the chemical reaction. Remember? So, what must happen before a chemical reaction can start? Let's see the options. Option number A is the temperature of the reaction mixture must decrease. B is the energy level of the reactant molecule must be below the activation energy. C is the energy level of the reactant molecules must reach the activation energy. And D is the concentration of reactants must be increased. So what must happen before a chemical reaction can start? So for a chemical reaction to start, the reactants, I have shown you the energy diagram, the reactants need to reach a certain stage of energy after which they can get converted into product. I have told you about the transition state theory also. So that particular, to reach that particular energy, that difference is called the activation energy. So basically the energy level of the reactant molecules must reach the activation energy. Okay, if you decrease that, the first option, temperature of the reaction mixture must decrease. If you decrease the temperature of the reaction mixture, the kinetic energy will be decreased for the molecules, and the chances of molecules getting colliding with each other and the reaction to proceed will get lowered. Okay, so in order to make sure that your reaction proceeds towards the forward direction, you have to make sure that the temperature is not decreased. Energy level of the reactant molecules must be below the activation energy. If the energy level of reactants are below the activation level of the reactant molecules is below the activation energy, it will never cross that energy barrier. So activation energy is kind of a hill, and for a reaction to reach till the product, they have to cross that hill. Okay, so if it is below that hill, that can never jump over that hill. So it has to reach the top of the hill in order to jump to the other side of the hill. Okay, so option number C is the correct answer. So option number C, the energy level of the reactant molecules must reach the activation energy. This is the correct answer for this question. So moving to the next question. Intermediate compound theory is proposed for. So we have four options: heterogeneous catalysis, B is enzyme catalysis, that is biocatalysis, C is micellar catalysis, and D is homogeneous catalysis. So I have told you about heterogeneous catalysis. What was the theory of heterogeneous catalysis? I have discussed right now. What was the theory that explains the mechanism of heterogeneous catalysis? We discussed the question, like it was question number 16 or 15, I guess. Yes, adsorption theory. Adsorption theory explains the heterogeneous catalysis. Okay. So similarly, intermediate compound theory is the theory that is proposed to explain the mechanism of homogeneous catalysis. Okay. So these two catalysis mechanism is very important. Enzyme catalysis is also important in terms of biology. Answer should be answer is D, option number D, Chetna. It's not option number B, it's option number D, homogeneous catalysis. So what happens in the intermediate compound theory, I'll explain. So in a homogeneous catalysis, there's no solid surface or a liquid, like there's no difference in the physical states of the molecules, right? The reactants and the catalyst all are in same state. So there's no point of explaining the mechanism using adsorption theory or absorption theory. So in this case, what is the proposed mechanism for homogeneous catalysis? It is intermediate compound theory, where the catalyst combines with one of the reactants and forms an unstable reactive compound called intermediate. So what happens? The catalyst, now I have told you that the catalyst never participates in the reaction, it just helps the reaction. So what happens is, the catalyst in the first step, it combines with the reactant and forms a unstable reactive compound. It is called intermediate. And then in the second step, that intermediate will react with the other reactant. So let's say it's a bimolecular reaction. If it is a bimolecular reaction, you have two reactants. Okay. So one reactant will react with the catalyst first to form some X compound, which is the intermediate. Then that intermediate will react with the other reactant to yield the product, and the catalyst will get released. So in this way, the catalyst will be renewed. In case of adsorption theory, we saw that the platinum surface was getting freed after the reaction was complete. Okay, after the hydrogenation of ethylene was happening, the catalyst surface, the platinum surface was getting free. Similarly, in this case also, we need to replenish the catalyst, because catalyst is never consumed in a reaction. This is a very important point you have to same happens with the enzyme catalyzed reaction. Yes, you are right in that way, like, but in that case there is no intermediate form. If you see, in enzyme catalyzed reaction, you have a protein which has a catalytic site, and your substrate will sit on that catalytic pocket and get converted into product and get released. So it's more similar to the adsorption theory than the intermediate theory, if you want to see the similarity. So we can't say adsorption because it's a protein and a molecule interaction. It's not a, if the substrate never gets adsorbed to the protein, that enzyme which is a protein. Okay, so you have an enzyme has a catalytic pocket. Your substrate will sit in that catalytic pocket and gets converted into the product and get released out of the catalytic pocket, and then again the protein is free for the next molecule of substrate to uh, sit inside that enzyme. Okay, did you understand the difference, Chetna? In many reactions, enzymes are covalently modified. Yeah, but then the enzyme will, uh, in that case also, you are not getting intermediate compounds. So, if you see in this example of homogeneous catalysis, if you see in this example of homogeneous catalysis, we have the reaction is SO2 plus O2 goes to wait SO2 plus O2 goes to SO3, 2SO3. Okay, so NO, nitrogen oxide, is the catalyst here, and what happens to the nitrogen oxide? Nitrogen oxide first reacts with the O2 to form NO2, and then NO2 will react to the second reactant, that is SO2, to form SO3 plus 2NO. So nitrogen oxide is again regenerated. Okay, but in what you are saying, in case of enzyme catalysis, even if the enzymes are covalently modified, the enzymes are like that can't, that is not a reaction intermediate, because the enzyme, that covalently modified enzyme, is not reacting with another molecule of reactant. Okay, I hope I am clear. So in an enzyme catalyzed reaction, it's just the substrate interacting with the enzyme, and the enzyme just catalyzes the changes in the substrate in order to make, convert it into product. And once the substrate is converted into the product, the dissociation constant is like the dissociation energy is higher for the product and the enzyme, so the product gets released out of the enzyme's catalytic pocket. In this process, even if a enzyme is getting modified while converting a substrate into the product, that modified enzyme is not uh, interacting with another molecule of substrate in order to make the product. Okay, in this case, whereas the NO2, which is the intermediate, it is interacting with the another reactant, that is SO2, to form the product. Okay, so in this case, if you see these two reactions, NO plus O2 gives NO2 and SO2 plus NO2 gives SO3 plus NO. If you add up these two reactions, you will get the first reaction. If you add these two reactions, you will get 2SO2 plus O2. So 2NO2 and 2NO2 will get cancelled out from the first and second row, and 2NO will get cancelled out from LHS of the first row to the RHS of the second row. So you will get 2SO2 plus O2 gives 2SO3. Understood? Is that clear for you, Chetna? Yeah. So you are you were right in case of enzyme catalyzed reaction. The like enzymes also catalyze the reactions in the same way. The enzymes uh, convert the substrate into product and gets regenerated, but in that case of regeneration there is no intermediate form, but in this case of regeneration one another molecule is formed. So sometimes in such types of reactions, let's say,
Say,
sorry, hello, yeah, so let's say we have how do I explain, yeah, so let's say this NO2 is an important compound that you want. So what people do in these kind of intermediate driven reactions, people they add certain substances so the reaction stops at this intermediate stage and they can get the intermediate out of the reaction scheme. Okay, but in case of enzyme catalyzed reaction that won't happen. You won't to you will either you can get a substrate bound enzyme or a free enzyme. There is no intermediate in that case. So that's why that theory does not apply to biocatalysis, it applies to homogeneous catalysis. And adsorption theory, so that enzyme catalysis is actually similar to the adsorption theory, just in that case adsorption is not happening. In that case, yes, intermediates a transient occurrence in case of biocatalysis. You are correct. Yes. So that's why it is different from intermediate compound theory. Okay. So this was the last question of this session. Is there any other doubt in any of the question, or you want me to show any other question again? If you want me to show any other question, I can explain it. Or if there's any other doubt regarding this 4th week session. So mainly in this session we have to remember the concepts of different types of catalysis: heterogeneous catalysis, homogeneous catalysis, biocatalysis or enzyme catalysis, micellar catalysis, and then different types of catalysts that are present. So positive catalyst, negative catalyst, autocatalyst and induced catalyst. So you should know the at least one examples for each type of catalysis and the theory behind, like the principle, the definition of what is what. So positive catalyst means it is increasing the rate, negative catalyst means it is decreasing the rate, then induced catalyst means it is inducing the reaction, autocatalyst means something which is formed as a product will catalyze its own reaction. So something like you should know at least one statement definition of each type of catalysis. And then there are two other terms, catalyst poison and catalyst promoters. So promoters of catalyst are basically something that can enhance the activity of catalyst. So in terms of biocatalysis, it is a very important concept because in biocatalysis, the catalysts are the enzymes. So all the enzyme inhibitors and enzyme activators, those are basically the catalyst poison and the catalyst promoters respectively. So in biology, it's a very important term, enzyme catalyst, catalyst poison and the catalyst promoter.